My topic is biodiversity and conservation of papaya. I am thankful to Dr. Prashant Bakshi sir, Associate Professor Division of Food Science SKUAST, for giving us this wonderful platform for making our presentation more expressive. Presented by Richa Sharma. Let's begin. A very warm welcome to all of you. This image depicts seedling, bud, female flower and male flower of papaya. Unripe fruit of papaya is source of papain and yellowish pigment present in ripe fruit is caricaxanthin. Papaya is a tropical fruit sensitive to strong winds, frost and highly susceptible to waterlogging or stagnation. Papaya is usually dioecious, but hermaphrodite and gynodioecious types are also recognized. It is highly remunerative crop. Among fruits, papaya is second rich in vitamin A after mango. Type of fruit is large hollow berry. Type of inflorescence is axillary panicle, highly cross-pollinated crop, and the pollinator of papaya is wind. Papaya seed is enclosed with gelatinous layer sarcotesta. Biodiversity and conservation of papaya. Biodiversity. It is a sum of genes, species, and ecosystem in a region. Types of biodiversity. The biodiversity is of three types: genetic diversity, species diversity, and ecosystem diversity. Genetic diversity, it refers to the variety of genes and genotype found in a particular species. Species diversity, it refers to the sum total of species found in a region. Ecosystem diversity, it refers to the variety of biological organisms found in various ecosystems. Advantages of biodiversity. It provides protection from biotic stress. It provides protection from abiotic stress. It provides choice for hybridization, adaptation to environmental changes. It provides genetic base in a breeding population. Conservation of biodiversity. To avoid danger of uniformity by avoiding outbreak of disease and pest. To provide food security to the future generations of people. To reduce pesticide use. To achieve further increase in productivity to feed increasing world population. To achieve further improvement in quality character of different crop plants. Conservation methods. There are two types of conservation methods: in situ conservation and ex situ conservation. In situ conservation is further divided into national park, on-farm conservation, and biosphere reserve. Ex situ conservation is further divided into two: whole plant conservation and parts of plant conservation. Whole plant conservation includes field gene bank, botanical garden, and parts of the plant includes seed gene bank, pollen bank, tissue culture, and cryobanks. Cryopreservation is one of the most important components in cryobiology. Cryopreservation is a viable freezing of biological material and their subsequent storage at ultra-low temperature, that is -196 degrees Celsius, in liquid nitrogen and nitrogen vapor. Steps in cryopreservation: selection of plant material, pre-growth, addition of cryoprotectant. Freezing is further divided into three: rapid freezing, slow freezing, and stepwise freezing. Storage, and finally thawing. Geographical origin and distribution. Carica papaya is believed to have originated in the lowlands of eastern Central America from Mexico to Panama. It was introduced to India during the early part of 16th century from Philippines through Malaysia, and it has only one species of papaya, namely Carica papaya. Distribution of papaya. Caricaceae consists of 6 relatively small genera with 35 species, all of which are American except Cyclicomorpha, which is from West Africa. Varietal testing in papaya is carried out under the All India Coordinated Research Programme of Fruits. Research and evaluation is carried out in various institutes locating in different agroclimatic zones. Was initiated during the 4th-5th year plan. Testing is carried out both by state agriculture universities and ICR research institutes coordinately to find solutions to various problems and to evaluate the suitability, adaptability, and transferability of the technology. Papaya is a popular tropical fruit crop. However, because of extensive monoculture and a narrow gene pool, it is susceptible to numerous diseases, including papaya ring spot virus, which has caused widespread damage in many countries. Conservation of the germplasm of the crop gene pool is important to provide a source of genes for the future development of resistant varieties, either by genetic manipulation or the conventional plant breeding. Papaya seeds generally classified as having intermediate seed storage capacities. Two procedures, desiccation and cryopreservation of seeds and vitrification-based cryopreservation of shoot tips, have been employed, and growth of regenerated plants have been studied. From here, it is concluded that cryopreservation of shoot tips and seeds of papaya can be considered for maintenance of papaya germplasm. Work is being undertaken to further optimize these techniques. Papaya seeds has been stored for 12 months at a suitable range of moisture content and temperature including cryostorage and was germinated after GA3 treatment. Germplasm inventory. Germplasm inventory management system records seed flow within germplasm banks, breeding programs, and research institutions that develop germplasm. Its main function is to manage information about germplasm quantities and to track germplasm history, the process of germplasm production over successive generations. It is defined as the legal enforcement of the measures aimed to prevent pests from spreading or to prevent them from multiplying further in case already they have gained entry and have established in new restricted areas. Main activities are plant quarantine, tracing, identification and control of quarantine pests, pest risk analysis, evaluation, testing and determination of quarantine conditions for imported and transit plant shipments. The significance of plant quarantine has increased in view of globalization and liberalization in international trade of plant and plant material in the wake of sanitary and phytosanitary agreement under WTO, prevent the introduction and spread of exotic pests that are destructive to crops by regulating or restricting the import of plant or plant product. Plant quarantine is vital to prevent the introduction of non-indigenous pests and diseases into a country or to intercept and eradicate them before they can be widespread and well established. By not following properly, plant quarantine resulted in outbreak of Paracoccus marginatus in papaya fruit. It is a mealybug native to Central America. It had spread accidentally outside its native range by trade in live plant material such as papaya fruit. It is a polyphagous insect and spreads rapidly, having infestation on aerial plant parts of papaya. First of all reported in Tamil Nadu. In Jammu Kashmir, it is introduced in the year 2009. Intellectual property rights. These are the legal rights granted by government authorities to control certain products of human intellectual effect. It includes patents, copyrights, trademarks, plant variety rights, geographical indication, and some jurisdiction trade secrets. IPR status of papaya. For example, papaya ring spot virus sequences and court-mediated resistance is a US patented. Geographical indication is a name or sign used on certain products which corresponds to a specific geographical location or origin. The use of a GI may act as a certification that the product possesses certain qualities or enjoy a certain reputation, essentially attributable to their geographical origin. A geographical indication registered with All India jurisdiction operate as per the Geographical Indication of Goods Act 1999. The geographical indication of papaya is Andhra Pradesh. GIS has given a role in analyzing potential and current distribution of target species, locating and assessing the population of target plant species and assemblages, measuring biodiversity, monitoring biodiversity patterns, and identifying priorities for conservation and management. When-borne pollen dispersion of papaya was studied. Documentation process involves information gathered from individuals through detailed questionnaire, focus group discussion with person having knowledge and published secondary information. Every effort should be made to identify the person with proven knowledge of local biodiversity. Process of people biodiversity register preparation, formation of biodiversity management committee, sensitization of the public about the study, survey and possible management, training of members in identification and collection of data on biological resources and traditional knowledge, collection of data, analysis and validation of data in consultation with technical support group, preparation of people biodiversity register, computerization of information and resources. Thank you.